Assalamu alaikum, dear listeners. The topic for the today's discussion is gene cloning through recombinant DNA technology. Gene cloning is a process of producing multiple copies of a gene. This can be achieved by PCR, polymerase chain reaction, and recombinant DNA technology. PCR is an ex vivo process, while the recombinant DNA technology is an in vivo process that involves the use of the bacterial cells. The main components of the gene cloning through recombinant DNA technology are gene of interest, molecular scissors, vectors, molecular glue, and the expression system. Gene of interest. Gene of interest, or also called as the gene of the desire or gene of choice, is the gene that is to be copied. This can be obtained in three ways. Small size genes can also be synthesized artificially using artificial DNA synthesizers, but this method is costly. Reverse transcription. A gene of choice or gene of desire can also be obtained by the reverse transcription process of a messenger RNA into the cDNA. Gene of choice can also be isolated from a living cell. First of all, the genome is isolated, and then the gene of choice is cleaved from the genome using the restriction enzymes. Molecular scissors or the restriction enzymes. Molecular scissors are also called as the restriction enzymes or restriction endonucleases. These enzymes are obtained from the bacterial cells. These enzymes are called as the restriction enzymes because they make a specific cut at a specific site called as the recognition or restriction sites. These enzymes cleaves the phosphodiester linkage of both the DNA strands. In recombinant DNA technology, the same restriction enzyme is used for cleaving the vector and isolation of the gene of choice. Vectors. Vectors are also called as the molecular carriers. These are usually the DNA molecules that carry the gene of choice from the donors to the recipients. In recombinant DNA technology, the small size vectors are more desirable because they can carry a large size genes. Some of the common vectors include plasmid cloning vectors, bacteriophage cloning vectors, cosmid cloning vectors, bacterial artificial chromosomes, and yeast artificial chromosomes. Molecular glue. Molecular glue is an enzyme called as the ligase. Ligase, they help in the synthesis of a phosphodiester linkage, and this results in the formation of a recombinant DNA. A phosphodiester bond formation occurs at the terminals of gene of choice. Expression system. Expression system is usually a suitable host to replicate the recombinant DNA. The most commonly used expression systems are the bacterial cells. Expression system can be used for the production of the specific proteins from the gene of choice and also for the production of the copies of genes. Protocol. The recombinant DNA involves the isolation of the genome from the host. From the genome, the desired gene is isolated using the restriction enzymes. The fragments produced by the action of the restriction enzymes are called as the restriction fragments. Each of the restriction fragment has sticky ends. The same restriction enzyme is used to cut off the vector. The gene of choice is inserted into the cut open vectors. Ligase enzymes synthesize the terminal phosphodiester bonds, and the recombinant DNA molecules are inserted into the expression systems. Thank you. Like and subscribe.